Good day. My name is Gerhard van der Horst. I'm a member of the Comparative Spermatology Laboratory at the University of the Western Cape, and I'm going to present this webinar in conjunction with my colleague, Dr. Liana Maree, who will introduce herself.
Hi, I'm a senior lecturer in the Department of Medical Bioscience, and I'm in charge of the Comparative Spermatology Lab. We're a nice, vibrant research group studying the sperm biology of honeybee till human. For this webinar, we are going to focus on two components that is critical in terms of sperm functional testing. First of all, we're going to look at the basics of sperm functionality and focusing on motility subpopulations, fragmentation, vitality, and mitochondrial membrane potential.
We will also focus on those specific sperm functionalities that relate to the female reproductive system. And I will rather show you in the next slide what these entail, amongst others: sperm cervical mucous penetration, capacitation, which is which one of the endpoints, hyperactivation is an important endpoint, the acrosome reaction, sperm zona oocyte binding and interactions. And here you can see the actual areas in the female reproductive tract where this may happen and where it actually presents the challenge to the sperm. Here, of course, the cervix with the sperm cervical mucous penetration, and then the rest actually occurs in the fallopian tube, such as capacitation, hyperactivation, acrosome reaction, and so forth. And so these are really the aspects where one needs to develop tests that actually relate to these important aspects of sperm in the female reproductive tract.
Gerhard, why do you think both these approaches are important?
Yes, I think it's extremely important on the one hand just to consider the basics of sperm physiology. A simple thing like sperm motility tells us a great deal about the function of the sperm. It tells us that the sperm membrane is intact, that the sperm is probably a fairly normal form, that metabolism occurring inside the sperm, making ATP and so forth, that all of that is really intact. And so while sperm motility measurement, particularly through a CASA system, such as SCA, is extremely important in terms of quantifying it and telling us about the basics. On the other hand, we now need to apply these in the sperm functional context, and that is why we need to develop sperm functional tests. So I again need to ask you, what do you see as the clinical importance of these?
So many of these sperm functional assessments that we do and the parameters that we measure have been found to be clinically relevant in terms of correlations with clinical pregnancy as well as a live birth outcome.
Thank you very much. Okay, let's then start straight away with the basics of sperm function. 5 components will be discussed here, from the subpopulations of sperm motility, the mitochondrial membrane potential, vitality, hypo-osmotic swelling, DNA fragmentation, and then, of course, the question of the centrioles, which is new to most of you. So, this is typically how a semen sample would look like and a positive phase contrast using the CASA system of MICROPTIC. And you can see quite a dirty sample. However, when it's analyzed, it shows the tracks beautifully in terms of the rapid tracks in red, rapid progressive, the green tracks less progressive, and then the blue ones really non-progressive tracks. So how do we prepare sperm now to look at these different subpopulations, which I've alluded to and which is very important? So there are many methods, and I'm not going to go into detail here. The classical method is the swim up, where we put a raw semen sample down at the bottom. The sperm swim up in the medium, and the sperm is captured here. Or you can do a differential centrifugation, whereby you use colloidal solutions of 45 and 90%, and you put raw semen here, you centrifuge it for about 20 minutes at 300 G, and then you collect a very motile pellet at this point. And then you can do various things with it. And we will allude to this question of the flush technique later on. So at least now you sit with different populations, and the question is, what are actually important here? Just simply the importance. It's not important to look at averages as a whole, but really averages of the subpopulations. And we can ask ourselves, of course, which ones. And yes, we can look at percentage groupings. We can look similarly at the progressive groupings of the kinematics, and also just simply for a rapid, medium, and slow moving sperm. This is quite getting a busy slide, and I don't want to go into the details except to show you that these things are quantified extremely well within the CASA system, and here in relation to percentages, but here also in terms of, for example, the kinematics. And you can see for all 8 kinematics, the various groupings of progressive, rapid progressive, medium progressive, all these values are given. And this is then very useful to actually look at these populations and see, do they indeed differ? Are they really the important ones? We think they are. Mitochondrial membrane potential relates very, very closely, of course, to motility. And I want to allude to 2 techniques. There are many, many ones. This one is the diaminobenzidine test. And you can see in human here, when there is very good mitochondrial activity, it shows up really well here in terms of green, here in the boar. And also, if the mitochondrial activity is poor, then there will be less activity here and less staining here. The advantage of this technique is that you only need phase contrast microscopy. Perhaps a more detailed technique is mitochondrial membrane potential, as done by MitoTracker Red, and you can see how well it defines the mitochondria within the midpiece. And this can also be used for flow cytometry. So very important to allude to what is actually the mitochondrial activity. Then hypo-osmotic swelling combined with vitality, a very useful test, which is a fairly new one, used called the Bright-Witt method, and it does not only distinguish between dead sperm, which stains pink or red, and white sperm, which is not stained, but also shows swelling of the tail. And as you may well know, that tail swelling, like hypo-osmotic swelling of the tail, actually shows that you're dealing with a very high quality sperm. It has the ability to swell. So this is the advantage of measuring 2 important functional aspects of sperm. And DNA fragmentation is very important because it can lead to problems in terms of fertilization, and also may in severe cases lead to cause actually miscarriage. And here are actually 2 tests which are commonly used. The halosperm test, and you can see a fragmented sperm here is being circled by red. The system analyzed it correctly as fragmented. These ones have been exposed to fragmentation medium, and so they're not really fragmented, but they only fragment because we do it artificially. And so it distinguishes very nicely between the sperm that is really fragmented and those ones that are not fragmented, and express it as a percentage. And you can see for halosperm, it needs to be below 28%. The toluidine blue test shows these big purple cells as fragmented and the very light blue ones as not. Extremely easy test, very useful, and very close relationship with tunnel. And just the final test here, which is not fragmentation, but deals with the nuclear maturity. And so in that one, there's not proteins yet, it's still histones, and it stains accordingly with aniline. Finally, I just want to allude very briefly to the importance of the centrioles, and that they are becoming more and more important. These are not only important in terms of structural components of the sperm, but also in terms of the fact that they are very important in terms of fertilization itself. and in terms of for the pronuclei of both the oocyte and the sperm cell to actually meet. And this is the wonderful work of Tomar Avidor-Rice's lab in Toledo, where they've actually managed to show exactly which proteins these things consist of and how they interact at various stages. And this may become a really important test in terms of measuring what we think can become very important in some cases of idiopathic infertility. So this brings us then to the end of this part on the basics of sperm function. In the previous section, I have alluded to some of the basics of the sperm functional tests, particularly those that relate to the subpopulations in terms of spermatility, etc. I want to ask my colleague now, why do you consider it important that we study subpopulations of sperm, particularly as related to spermatility?
Well, as we know, the semen actually consists of a heterogeneous population of sperm. And apart from the highlights you've given in terms of how you evaluate the CASA subpopulations, it's also important in the clinics when they separate sperm into different sperm fractions, for instance, a high motile and a low motile fraction. And then to assess these populations independently, what we've done in our lab is to study these subpopulations, and we found large differences between a high motile and a low motile sperm population, not just in terms of motility, but also all the other sperm functional parameters that we're discussing in this webinar, and also their relationship to the original semen characteristics.
Okay, and all of that would then assist to give a better understanding and outcome if you use the best population in terms of your IVF work. And I'm actually wondering, and the people listening to us tonight, I'm just asking you, you know, are you working with subpopulations? You've heard it's important. We think you should. Thank you. So let us start now with the sperm functional tests. And first of all, the sperm cervical mucus penetration or sperm mucus penetration test. And I've distinguished here very clearly between the classical sperm cervical mucus penetration. The rationale here is that we actually follow sperm progression inside cervical mucus, which have been obtained from donors. And secondly, a more modern test, the sperm mucus penetration, where the sperm is challenged by its own seminal plasma. So let us see what happens here in this instance, where we have a typical analysis of sperm motility in seminal plasma. And here you can see how the sperm is actually swimming, progressing. And what we do now is we look at the analysis of which sperm are actually past the test of mucus penetration. And if the sperm actually managed to show a VAP above 25 micrometers per second, a straightness above 80%, and an ALH above 2 but less than 7.5, then according to these parameters, they have actually passed this mucus penetration test. And in this particular software of MICROPTIC, it puts blocks around those sperm which actually passed it. What about the interpretation of the results? So this is very important. And you know, we don't even need to know the sperm concentration, but we need to know the sperm volume, because it expresses the total number of sperm that passed the test, the mucus penetration test according to those parameters, in the total semen sample. And it needs to be, for a good quality sample, above 5 million for the ejaculate, and you can see it is actually 7.9 million. So in this instance, it's a happy chappy as far as this sperm functional test is concerned. And this brings us then to the end of this test.
Gerhard, in terms of this test, can you maybe tell us more about the pitfalls that is important here, as well as why do you use seminal plasma instead of cervical mucus?
Yes, I think it's a really relevant question, and I think in the first instance, of course, the ideal is to use cervical mucus, but it's very difficult to sample except in a clinic where you have many gynecologists who can get permission and sample and get samples of cervical mucus. But I think everybody listening will agree, and I think you will agree with me, it's extremely difficult that you're going to have a constancy in it, that it be consistent rather. And so what do you measure? If you now use it and you've stored a lot of these in the fridge, and every day you use it, you're not comparing the same thing. So Dr. David and Dr. Sharon Mortimer came forward with the ingenious idea of using the seminal plasma of the person who you are testing. In other words, you are actually testing it in his semen, and it's challenging the sperm in that environment. It's actually an excellent way to do it. And also the viscosity of, in most instances, of seminal plasma is very, very much the same as cervical mucus. And so it is a much better test, almost having its built-in control. Yes, so I think that is why it is becoming a very good test, and there are very specific cutoff values. For example, in the sperm class analyzer of MICROPTIC, we use very specific kinematic cutoff points so that you can actually quantify it very accurately.
Wonderful, thank you.
So here we start with the second and probably most important sperm functional test, namely hyperactivation. But let's first consider the processes that lead up to hyperactivation. We refer to this as sperm capacitation, and it involves a whole range of processes, and I'm not going to go into detail, but I will show some of them. It removes molecules from the sperm membrane. There's increased respiration, and there's changes in lectin binding, and particularly calcium accumulation and increased cyclic AMP. And the end product of capacitation is hyperactivation. And let us now just review the important aspects of hyperactivation for human sperm. As indicated, it shows that there is evidence that capacitation has taken place. And the next point is one that very few people realize, and that is that it has a dual function of attachment and detachment from the epithelium of the oviduct. In other words, it's not just to become hyperactivated to attach to the oocyte in the end and to penetrate the zona, but it first needs to land into the oviduct, and then it very quickly attaches to an oviductal cell. And it can only attach to it if it was hyperactivated. And once the oocyte arrives, it detaches from that epithelium and then follows these processes. And we know that it relates to fertilization and live birth outcome in humans. And we need an hyperactivation of more than 20%. And these are some of the kinematics within the CASA program that can help us to determine this. In order to be able to do this in the laboratory, in other words, to challenge sperm and see functionally how good they are, these are a whole range of substances such as caffeine and progesterone that will all induce hyperactivation. I showed the slide previously, and I indicated that once we have provided some very nice sperm, we can either use it here in a very interesting way, where we first provide in a layer slide raw semen or washed sperm, and then we displace it with that medium containing the agent for hyperactivation such as caffeine. And we will then, because the sperm has now been displaced and this is the new medium, the sperm will swim into it and it will then be either hyperactivated or not. And here is a beautiful example of sperm in phosphate buffered saline, but then using the flush technique, using 5 millimolar procaine hydrochloride causing vast hyperactivation. This is human sperm in 5 millimolar caffeine, and all these sperm are highly hyperactivated. In other words, they have a VCL above 150, a linearity less than 50, and an ALH above 70. Let's have a look at the detail of a sperm like that. And here you can see a star sperm pattern, which is very typical of hyperactivation. This is the kind of sperm that we imagine would be able to fertilize the oocyte. And of course, there are transitional phases, but these are all full hyperactivation or moving to star sperm hyperactivation because they're exceeding these parameters or less than 50% linearity, and are therefore being able to be called hyperactivated. And here are some of the forms. This is how it looks like within the CASA program of the SCA MICROPTIC. And you can see we have these beautiful patterns, and these are each one representing 1/50th of a second. You can see 1, 2, 3, 4. Every time we represent it as in this instance as a 50th of a second. And here, Van Horst and Sanchez has developed a model to show the pattern of sperm motion in 3D. So this is the actual 2D pattern, and this is with in taking into account the potential 3D using the Z value. And in really star sperm hyperactivation, this is how we imagine how this would look like. And it really assists us just to understand maybe the potential pattern of hyperactivation. So we will now discuss the importance of hyperactivation in the next session.
Thanks for the explanation of how hyperactivation is measured. Can you maybe highlight the key problems with the standardization of CASA in terms of hyperactivation?
The problem is that different manufacturers of CASA instruments have different ways in measuring hyperactivation. And probably they come out at more or less the same answer, but not the same. So there is unfortunately poor standardization. For example, in the Hamilton-Thorne system, the Mortimers have suggested they use the combination of curvilinear velocity and a d-fractal, which seems to be very accurate. For most other CASA systems, it works very well to use other means. For example, in the MICROPTIC SCA CASA system, we still use curvilinear velocity cutoff point, as I've shown. We use linearity cutoff point, and we use ALH. But there are numerous problems associated with this. It all depends how you measure the average path velocity. And that is really the key problem. If we can standardize that somehow, then we can measure ALH accurately, and then we may have a better assessment. So those are two of the facets which I think is relevant.
Thank you.
Yes, and of course, we know hyperactivation being so incredibly important sperm functional test. Can you just allude to the clinical applications?
Yes, of course. Already about 3 decades ago, it has been indicated that a sperm semen sample that have about 20% hyperactivation is has a good fertilizing potential. And interestingly, we found that in most mammals that we've studied as well. So 20% seems to be a good cutoff. So if you now have your sperm population and you measure and it gets to about 8%, you know there's going to be a problem, maybe with hyperactivation in the female reproductive tract.
Thank you very much. And now just to come back to my initial issue of if it's been indicated, for example, that if you use the fractal approach, that your hyperactivation outcome is at least 25% higher than the others. So yes, on the one hand, you've shown the clinical application, but we need to standardize it. The last sperm functional test that we will deal with tonight is the acrosome intactness and acrosome reaction aspects. And two aspects are really relevant. I think it's very important that one must realize that sperm needs to have an intact acrosome, but moreover, there needs to be at least more than 40% cover of the sperm head. Then, when we perform the acrosome reaction artificially by using calcium ionophore, there needs to be at least a 15% difference between control versus the induced sample. The rationale and how we do it is briefly the following. Peanut agglutinin attaches readily to the acrosome and is used then as a fluorescent stain. And we use a FITC filter. And one can readily see blue-green fluorescence if acrosome is present and quantified using the SEA acrosome module. So, here is the methodology that we have used for the SEA acrosome reaction, and we've developed it as a quantitative CASA test, and we've combined the use of Horst and the lectin PNA. And in this instance, the acrosomes will intact acrosomes will be bright green to blue fluorescence, whereas if there's no acrosome and there's an acrosome reacted, it will show blue. And here is an example where we only use PNA, and you can see the test won't work with that only, because you see these beautiful acrosomes, but you've actually, if you look at phase contrast, you've missed out on those two. And so one needs to employ Horst also, so that the rest of the head is stained blue, so that when the acrosome is reacted, you will at least see the blue stain. And here is an example of acrosome intactness and reacted, where you can see this is the sample and this is how the CASA system analyzes it, and it's recognized all of these as acrosome intact, and of course that one, yes, as reacted. But now, more important is just as not only intact and reacted sperm, but what happens if we apply the calcium ionophore. In other words, we induce the acrosome reaction. And ideally, there needs to be a 15% difference between the control and the acrosome reacted sperm. And you can clearly see how well the system recognizes those that are acrosome reacted. I indicated it right at the beginning that yes, it is fine to say that we have intact acrosomes or reacted acrosomes, but what if those acrosomes are abnormal, like in these instances, where there is clearly very small acrosomes? So, of course, they need to be of a normal morphological size too. So, I think we need to take this also in consideration. We will now follow with a general discussion of all the sperm functional tests. So this brings us to the end of this webinar on sperm functionality. And the question now is, do we summarize a lot of things? Do we ask, so what? I think I just need to very briefly indicate that we're not going to summarize much on sperm biology, the basics. We've done that. You've seen it's important to work with a healthy sperm cell, happy sperm cell, and we will concentrate on the clinical facets. I'm going to ask Liana now to please allude to the importance in the andrology and clinical laboratories.
So we all know that in the clinical setup, the basic thing that they do is a standard semen analysis. But during the last decade or so, it's been proven over and over that a standard semen analysis is not enough, especially if there is unknown fertility issues, or if there is a subfertile patient. I think then it's very important that we start looking at these sperm functionality assessments and do that in addition to a normal standard semen analysis. And also want to highlight that we should consider using CASA as a tool for these assessments. We know CASA can give us very good objective assessment and also very reliable and repeatable results.
Fantastic. Yes, and also I think if I can just end off by saying we must realize in the old days, CASA was just motility, morphology, concentration. Now, in a system like MICROPTIC's SEA, it consists of a number of modules which actually quantify many of these functional aspects, like the vitality, there's the acrosome reaction, and so forth. So, yes, thank you for doing this webinar with me. I think it was a lot of fun, and I hope you have enjoyed it. And best of luck and many sperm for the future. Bye.
Bye.